Welcome to From the Spectrum podcast. This is a podcast about autism. It is my goal to explain what is autism. I plan to use a mixture of scientific literature, personal experience, and opinion. With opinion, I will explain why I feel the way I do and give examples. I will provide links to various references for each episode. For each episode, we will discuss various aspects of autism. For today's episode, we will explore autism's development in the embryo. We will cover common biology already discussed, such as cell proliferation, differentiation, migration, neurogenesis, synaptogenesis and synaptic function, and the development of neural networks. Think cells migrating to common cells that create whole brain regions, or organs, or tissues. The process of stem cell into a specialized cell, and how they migrate to each other to form different aspects of the human body. This episode will expand on the Cause of Autism episode to explain more detail. This episode fills in what is happening simultaneously and slightly downstream. The Cause of Autism episode explained how our living organism is created, at least an introduction to it. This episode picks up, essentially, the developed cell. And this is regardless of normal developed or abnormal developed. In addition, we will cover specific timelines of cell life and the embryo, and into about 3 years old, postnatal. Things to remember from Cause of Autism. Let's call them ingredients. Light, as the driving force of living organisms. Remember the energy source, the electromagnetic strip, the photoelectric effect, and water, magnetism, and the elements from the periodic table. Remember, we use some of these elements, and then a subset of the ones that we do use. A subset, we use a lot of those. But also remember melanin as the target for that light, and melanin being black to absorb all of the light spectrum. Don't think it's black for this. Think our body, our biology, our evolution chose melanin to do this role. Remember POMC as the source for melanocyte stimulating hormones, alpha, beta, and gamma. These are in cells, organs, and tissues, all through the body. Later, I will show you how this is proven as an implication to autism, accidentally, in a paper we will spend most of the episode reviewing. The paper proves light and melanin is a cause without saying it is a cause. Remember vitamin A and tryptophan and their roles in eye development. Eye development generates brain development. Vitamin D and tryptophan and the skin. TPH1 for the skin and gut, which helps create our body. And TPH2 for helping to develop the brain. TPH is tryptophan hydroxylase. And remember, proteins are semiconductors. And remember the amino acids. We have 20 amino acids. Tryptophan and methionine are codes for DNA and protein sequencing. Tryptophan and methionine are special, whereby they have one codon and they sequence proteins and DNA. Methionine is the special start signal. Tyrosine is huge too. Remember the aromatic amino acids. All of these so-called ingredients use certain wavelength of light. They require the certain wavelength of light to function. We've covered, and I would even say we've established, common themes in the biological development are implications to the cell and the synapses. These are common themes with autism. The cell, meaning proliferation, differentiation, migration, development, and the synapses, development, growth, and the functions. So let's explore something. Autism Spectrum Disorder Living Biology. This is the paper I want to review from Trends in Neuroscience. It is a paper from Cell Press. Recently, there's a conceptual and paradigm shifts towards a focus on the abnormal prenatal processes underlying autism within each individual, from specific abnormal development to early clinical stages. Remember, cells divide. They increase in number. So cell proliferation, increasing cells as a result of growth and division. Differentiation is becoming mature. Cells become specialized, unique or distinct types. These are stem cells can become nerve cells, which we mostly discuss in the podcast. They can become immune cells, muscle cells, organ cells, blood cells, epithelial cells, which is important in the light as a cause hypothesis. And melanin is inside all of these cells. Migration, once mature and specialized into one of those previous mentioned cell types, migration is like building blocks that allow a formation to occur, such as creating the nervous system, or an organ, tissues, or muscles, and so on. Let's review some timelines and the developmental milestones. First trimester, starting roughly at 4 weeks, proliferation and neurogenesis occurs, the beginning of neurons, or cells, lasting to around 26 weeks. This is prenatal. This is the embryo. Cell migration begins at 10 weeks or so, and this lasts until 32 weeks. The second trimester, Neurite overgrowth at 14 weeks to roughly 3 years of age. So, roughly at the end of the critical period that we've discussed quite frequently. And synaptogenesis and synapsis function begins at 20 weeks until around age 3. And the third trimester, neural networks, the formation and functioning. Once specialized, they start to begin these networks that develops the nervous system and so on. This starts at 32 weeks, and essentially, this is continuous through the lifespan. The paper distinguishes Epoch 1 as trimesters 1, 2, and 3, and Epoch 2, trimester 3. So trimester 3 is split between both epochs. Epoch 2 is trimester 3 into early postnatal periods. Let's call it that 3 years old. Epoch 2 is more brain specific, or if another type of health condition, implications to these areas occur. Remember, autism just shows up. It doesn't necessarily mean if you have a problem, it doesn't necessarily mean that problem will cause autism. It might cause something else, something similar. And this is the case with most health conditions, if we go that far upstream. For our topic, autism is a brain development, is a region of interest. This is covered in previous episodes, episode 3, 4, and 5, and in Autism and Light, the Cause of Autism episode. Disruptions here include Neurite overgrowth, synaptogenesis, and wiring, or connecting, those highways of communication. Remember I said highway, but some connections could be more like a gravel road. Remember myelin. Remember these connections differ. And this is how cells communicate. At each synapsis, a cell receives a brief summary about what must be occurring for this cell to connect here. With each connection, the summary adds and creates a story. Another implication, or expanding on the migration / brain development, because this is the process. These cells are migrating and creating the brain. Brain development and dysfunction with how our regions or networks form. Of course, that ought to make sense to us. However, the crucial part is the timing of it. Remember these episodes: Autism and Adaptive Responses, Autism and Salience Network. Remember our discussion with Dr. Liana Hernandez and the boy versus girl differences. Back to the Salience Network. Remember the UCLA paper came out in 2024. The paper I am mostly referencing about being able to predict profound autism. Remember the conversation with Dr. Catherine Lord, and I mentioned this. It has to do with the brain organoid. The paper predicting profound autism, and this paper from Trends in Neuroscience discussing the embryo, is from the same researcher. This researcher does wonderful work from UC San Diego. Okay, the Trends in Neuroscience paper. This paper mentions risk genes, a common topic with autism research. Specific risk genes are risk genes that we've covered on the atomic level and with general descriptions in mostly episodes 3, 4, and 5. These include phosphorus, phosphorylation, and P10, and protein kinase B. These involve cellular metabolism. Previously, I said autism and type 1 diabetes could be related upstream. These are the regions of interest. Diabetes will involve POMC too, a product cleaved from POMC for insulin. That's my thought. Remember, implications upstream look differently downstream. They have different so-called clinical presentations or different health complications. Now, I didn't say autism and type 1 diabetes are the same, or one increases the risk for the other. I'm saying upstream, many shared regions of interest are present. Anyway, I won't mention the trends of these each over the past 50 years, or even, let's say, the past 20 years. Again, at least not now. Also, mTOR is involved here in the paper. I mentioned possible hope with mTOR when originally discussing P10. A paper from Cell Press. This was in episode 3, at the beginning. Lastly, from the risk genes discussed, is a pathway that play a critical role with embryonic development. Everything mentioned, I believe, are components of the cell development. And this risk gene also contains mitogenic processes. Mitosis. This requires UV light. This is the spot to explore for autism. And the time is in the embryo. We've connected early findings postnatally at 6 weeks old. 2 studies. We've discussed 2 studies here. The Cellular network from UCLA and the brain organoids mentioned earlier, predicting profound or more severe autism from UC San Diego. I bring this up for 2 reasons. 1, to expand and prove light as a large possibility as the cause of autism. And 2, funding and research must go here and stay here until proven otherwise. This stuff on older children or adolescents, this research likely will not provide much to the world, at least not at this time. Research ought to move to the pregnant woman because at this point, every child seems to be at risk for autism. If we go by the 1 in 20 boys data and the possible misdiagnosed girls from the gender findings, it appears the assessment tools are boy biased because the diagnosis result is boy biased. But if girls or females have more social motivation that, of course, autistic boys and are similar or more social motivated based on brain imaging and connectivity than non-autistic girls and non-autistic boys, that data could be misleading too. Regardless, I don't want to discuss assessments now. The timeline ought to go to the embryo, and the cause is cell dysfunction in this development. Okay, let's discuss some specifics of abnormal brain development in the prenatal. These are post-mortem. Proliferation. These are averages. Autistic children have 67% more prefrontal neurons than non-autistic. This includes 79% more in the dorsolateral prefrontal cortex. Remember the subdivisions. The dorsolateral prefrontal cortex is involved with things like working memory and task switching, planning, inhibition, and, interestingly, abstract thinking. And a 29% increase in the medial prefrontal cortex, that subdivision that we discussed in autism and adaptive responses. Now you hear that there's an increase of neurons in these different subdivisions for autistics. But this does not mean that these areas are better. They function better. Remember the poor connections out of these areas. 30% more overall in the cerebral, and 57% more von Economo neurons or cells. These are spindle neurons. Remember from Autism and Adaptive Responses episode. There is an increase of these spindle neurons in the insula. Remember the insula, that hotspot, that regional hub that receives bodily information brain information and integrates it with how the living organism is navigating society, navigating the outside world. And the ACC. Remember in the adaptive responses episode, when we make healthy and adaptive responses to the flexible rules that are going on in socialness, going on in society, the outside world is very unpredictable. When we make healthy choices, the medial prefrontal cortex and the ACC lead the way. They lead those adaptive responsing. However, whenever we have trouble with responding to the environment or responding to our body, the insula kind of overtakes the prefrontal cortex and the ACC, and it kind of leads the way. It allows the body to overtake the brain, essentially. And these are specialized cells that are only found in larger primates. They're not across all species or even all mammals. These are involved in socio-emotional processes. So social-emotional processes. It sounds like an autistic phenotype description. A highlight of these data are this occurs between 10 and 20 weeks in the embryo, not postnatal. Okay, now a common theme with autism is excitation. The excitation inhibition balance, or imbalance. Cell proliferation regarding excitatory cells. A recent study exploring 102 common risk genes linked to autism. 71 of those 102 are involved in excitatory cells in the cortex. Add this to the data of inhibitory implications in the striatum and with 2 glia cells, oligodendrocytes and astrocytes. Remember the striatum, a hot spot for motivation and movement, motor movement. Also think the reward areas. Remember the discussion of the nucleus accumbens and then those stimulus responses. So here we just mentioned the ventral and the dorsal striatum. They are reflexes and deliberate, goal-directed activities here. These include socialness. These also include what is salient to us. And for autistics, what is salient is mostly staying within ourselves, within one or few interests at a time. Remember that biology that gives us autism allows us to be comfortable within ourselves. That's preferred. When I say motivation, it is not limited to deliberate wants or deliberate needs or likes. I think what motivates the nervous system to return to comfort. Those actions become the motivation. I know some discussion on genes, and I know it is a hot spot for research and funding. I don't love it. You heard Dr. Lord say, she is unsure if this is the way, what it will even do. My position is, I would rather spend time and money understanding the cause, such as the artificial light, over the genetic implications. I understand the genetics are implicated, the genes are implicated, the cells are dysfunctioned in various ways. But why? I suspect this is because medical treatment can be designed, developed, and sold at large volumes. Then profit. Profit is our medical paradigm. The most popular medications for autism. Yes, that's interesting, right? The most popular come from antipsychotic classification. These are antipsychotics given to autistic individuals. There are a couple of different things wrong here. One, the family, the loved ones of the autistic children, they're vulnerable. They want their child to have relief, to be able to fit in society. They want their pain and struggles to be reduced or eliminated. And the medical profession, with their training, is designed to prescribe medications. Try this. Try an antipsychotic or SSRI or an antidepressant. Because the child needs to interact more. The child needs more friends to be able to be comfortable interacting. The child needs a more diverse interest. They need more hobbies. The child receives complications in social settings whenever there is overstimulation. So let's try medication to calm the child, to calm the individual, so that they can fit in better and easier to be around and be accepted in society. I only bring this up because I have concerns. I have disagreements with medication use for autism, especially medications that were developed for other conditions. We mostly just throw medications at everything. Try it, and if it doesn't work, we'll try something else. And I'll tell you 2 reasons why it's concerning to me. It's very suspicious. One is, rarely do you get any informed consent. You just get the prescription, and maybe the physician or the PA or the NP, etc., will give you just a off the top contour, just a few side effects or a few implications that happen in a very questionable trial. The second thing is, the second concern, and probably even more concerning than the informed consent, is the medical paradigm, the accreditations. They cannot prescribe prescriptions fast enough. So over time, they've developed these nursing roles the role is to prescribe medications. They are trained, and their role is to prescribe medication management. Look at the health trends. Look around at society. Just observe each other. Let's go back to 1960, and then 1980, and year 2000, and 2022. And let's break down age from birth to 9 years old, to 10 to 19 years old, to 20 to 29 years old, so on until we get to 90 years old. Let's look at the number of prescriptions given in each decade of lifespan from the 60s, the 80s, the 2000s, 2020s. That increase is going to be wild. And nothing's changed. Nobody's healthier. Just today, Neuroscience News released, they highlighted an article. This says, cancer drug as a possible autism treatment. Okay, one, how is this even discovered? And what is going to be the purpose here? But two, more discerning is clicking on the link. It's not even autism. The cancer drug would help Rett syndrome, which has been kind of compared to autism, but as a separate thing. It's a syndrome. Syndromes mean not a lot is known about it or what to do about it. This came from UC San Diego as well. This was very disappointing, in my opinion. However, remember, autism just shows up in human biology, in human evolution. Something in the normal biological processes changed. That change appears to be light. And all of the new health trends supports both. both meaning the change and the profit. This specific paper from Trends in Neuroscience highlights autism as a multi-organ disorder. Of course, the primary one is the brain, meaning the biggest complication in autism is the brain. But this is huge. It is huge because of the light story and melanin in POMC. The paper highlights all of these separate organs implicated in the autistic phenotype, in the autistic individual. And there's a large list. Now, it doesn't mean that every one of these are implicated or abnormal, but there are a lot of shared implications throughout the body, which we've discussed. We've discussed in this episode even. What is going on upstream, those implications will show up differently, and sometimes in multiple ways. You've heard of sequela, or you've heard of a health condition causing other health conditions. All of these areas are dependent of melanin and the light to drive nonlinear optics. Amazingly, this is amazing to me. I was reviewing melanin and leukocytes. Just chalk it up to one of those B3 fixated interest. I'm just looking at melanin and where it is throughout the whole body. And I just happened to stumble upon this week. Melanin and leukocytes from the 1960s. This has already been known since the 1960s. How many people know about melanin being in white blood cells? And for those listening, leukocytes are white blood cells. How many researchers and scientists on autism know about melanin being in white blood cells? They know autism is linked to immune and inflammation. This is huge. These are huge findings. This proves, this suggests these immune problems and the inflammation problems, and we've discussed the mitochondria problems, and all of these cell implications and synaptogenesis and synaptic connections, all of these are kind of dysfunctioned. But now they're linking other bodily processes, other organs and tissues that are seemingly unrelated. This supports the light theory. Light is the only thing at this current moment that is making sense. The past 3 episodes, we've outlined some critical things here about the causes, about what's implicated in these causes, and understanding even the rates of diagnoses, and how much light has changed during this timeline since the 1930s. Not only has the light changed, but the amount of time and how humans have shifted behaviors more indoors under artificial light and surrounded by technology light, and less time outside. And this is showing up now in the lineage. Remember those timelines discussed previously. Remember that blue light chromophore, melanopsin. Remember it wasn't discovered until 1998, and then fully understood the sequencing until years after that, a couple of years after 1998, in the early 2000s. But now it's known to be across various aspects of the brain, deep inside the brain, in roughly 30 regions of the brain. It's in tissues and organs, epithelial cells. And remember, it's a chromophore, so it activates based off of specific light wavelength. And the melanopsin is around 480. It peaks at 480 nanometer, which is blue light. Modern light is blue light. It's in the shorter wavelength. And our shifts to this type of lighting runs parallel with the rates of autism. The timelines fit. Lastly, this blue light wavelength light shuts down. It implicates POMC, the pro-opioid melanocortin subdivision of the hypothalamus that cleaves many crucial parts of our biology, including the melanocyte-stimulating hormones. This is underrated in the research of autism. This ought to have attention, but it has no attention, and that is a concern to me. So I hope to help bring attention of autism and light, but not only just autism, the health implications because of how the human body uses light. If you are listening to the podcast or listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge. And I very much appreciate your feedback. You can contact me on X or Facebook or email info.fromthespectrum@gmail.com. Thank you for listening to From the Spectrum podcast.